Hi, everyone. Good morning, afternoon, evening from Singapore. I'm Anthony, a new assistant professor here at NUS. It's my pleasure to be the moderator today. So I'll be introducing two fantastic speakers from NUS. First, it's my pleasure to introduce Dr. Jiangbo Wei, a new assistant professor and a PYP fellow in the Department of Chemistry. Dr. Wei earned his bachelor's in chemistry from Peking University before pursuing his PhD and postdoctoral research in Chuan He's lab at the University of Chicago. He's an expert in RNA methylation and made significant contributions to the field with numerous publications. His recent work includes studies on how FMRP phosphorylation modulates neuronal translation through YDHDF1 and the role of FTO in line1 M6A methylation and chromatin regulation in mouse embryonic stem cells in development. Today, he'll be sharing his insights from his FTO-LINE1 RNA study. So please join me in welcoming Dr. Wei. Moving on, Mike, to you.
Thank you, Anthony, for the kind introduction, and thank you everyone for attending my talk. It's my pleasure to share my work on RNA modifications with you. Particularly, I will be talking about how FTO mediates line1 RNA M6A demethylation and consequently shape chromatin state and gene transcription. So first, let me briefly introduce the background. So we all know that gene regulation is a central factor in cellular form and function. The central dogma explains the process of gene expression. The genetic information is transcribed from DNA to mRNA and then translated to protein. The central dogma describes a sequence-based information flow. So beyond the primary sequence, DNA and histones can be chemically modified. These modifications are reversible and they dynamically control the gene expression, comprising the field of epigenetics. As we can clearly see here, mRNA is the epicenter of the genetic information flow. So what about mRNA? The earliest discovery of internal mRNA modifications can be traced back to 1974, when the presence of N6-methyladenosine, for short M6A, in mRNA was identified. However, whether mRNA modification can be reversible and also control the gene expression have been overlooked for decades. In 2010, the concept of RNA epigenetics was first proposed, and shortly after, this hypothesis was prompted by the discovery of the first RNA demethylase FTO. This study proves that mRNA-M6A is not a passive marker. It can be actively reversed and dynamically controlled. This revived the broad interest in the RNA field. In 2012, taking advantage of the high-throughput sequencing, the initial mapping of RNA-M6A was reported. Together, these milestone studies laid the foundation for the thrive of RNA epigenetics in the past decade. Notably, my advisor Chuan was awarded the Wolf Prize last year for his groundbreaking work in RNA modifications in gene expression regulation. So far, there are more than 170 different kinds of RNA modifications has been identified in almost all kinds of RNA species. Here, I listed a collection of mRNA modifications covering from 5' UTR coding sequence to 3' UTR. Among them, M6A is the most prevalent and well-studied mRNA internal modification. So before I dive into the details of M6A, let's take a little detour. I would like to show you one example of how RNA modifications are involved in our daily life. I believe most people here have received the mRNA vaccine against COVID-19. In the mRNA sequence of the vaccines from Pfizer and Moderna, all the canonical uridines are replaced by the 1-methylpseudouridine. This modified uridine would increase the mRNA stability and generally the protein production while reduce the immunogenicity. And of course, we all know the Nobel Prize last year. So going back to M6A, the writers, erasers, and readers, which respectively deposit, remove, and recognize RNA-M6A, are collectively known as M6A effectors. The majority of mRNA-M6A is installed by a methyltransferase complex with metal 3 and 14, forming the core catalytic center. FTO and RBH5 are the only two known erasers that can actively reverse the mRNA-M6A. The molecular functions of M6A are largely dependent on the reader proteins. For instance, YTHDC1 facilitates the splicing and nuclear export of methylated transcripts. YTHDF1 and YTHDF2, respectively promote the translation and degradation of their targets. Extensive studies have also highlighted the biological significance of RNA-MSCs in development and various human diseases. So here it looks like the writers and readers are enough to exert the function of MSCs, but it's not true. The biggest knowledge gap here is why nature evolves demethylases. They must be functionally important, otherwise it could be a waste of energy to install a mark on RNA and reverse it for nothing. So studies have shown that in response to signaling out stress, demethylases can be involved to erase MSCs in a subset of transcripts. The heat shock and UV damage studies are two examples. We also suspect that during cell state transition or developmental events, demethylase offers an option to fine-tune a subgroup of mESC sites rather than the global alteration by the methyltransferase. So with these questions and hypotheses, let me share the story today about the presence and functional relevance of mESC demethylase FTO and mESC demethylation during mammalian development. So as I just introduced, FTO was the first identified RNA demethylase. It can remove the methyl group of mRNA-MSCs. This demethylase activity was validated in vitro with synthetic single-stranded RNA probes and inside cells with mRNA purification. So since this initial report, extensive studies have shown that in many human cancer types, FTO can mediate mRNA-MSC demethylation of essential oncogenes or tumor suppressors to regulate cancer progression. In addition to human cancers, FTO has been long known to play critical roles in mammalian development. For example, FTO knockout mice display severe developmental defects. Most knockout mice died at the embryonic stage or shortly after birth. Those survived mice show significant growth retardation. Whether demethylation activity of FTO contributes to this in vivo phenotypes are unclear. We have been curious about the physiological substrate of FTO for a long time. We wondered if mRNA-MSC is responsible for these defects caused by FTO knockout. However, in a study of MSC mapping across human and mouse tissues, FTO mRNA expression actually shows slightly positive correlations with mRNA-MSC level. As an MSC demethylase, we would expect negative correlations. So these results indicate that generally in these tissues, mature mRNA may not be the primary substrate of FTO. So more recently, our lab reported that in mouse embryonic stem cells, MSC unchromatin-associated regulator RNAs could control chromatin state and transcription. We term these RNAs as CAR-RNAs, including enhancer RNAs, promoter-associated RNAs, and repeat RNAs. So MSC methyltransferase METL3 and the nuclear reader YTHDC1 are involved in this regulation. So importantly, in the genetic aspect, developmental phenotypes of FTO knockout mice are consistent with METL3 knockout or YTHDC1 knockout mice. These genetic similarities inspired us to explore the role of FTO in chromatin regulation. So the mouse embryonic stem cells offer an optimal system to study chromatin regulation and mouse development. So we derived FTO knockout and wild-type control mouse cells. We isolated the chromatin-associated RNA and consistent with the published tissue sequencing data. From our mass spectrometry results, FTO knockout showed almost no effect on mRNA-MSC, but significantly increased the MSC level of chromatin-associated RNAs. To study these changes in detail, we performed the MSC-MERIP sequencing with MSC-methylated probes as spec-in control. We found that FTO knockout leads to increased MSC levels of all three types of CAR-RNAs, and this increased MSCs were associated with reduced CAR-RNA expression levels. Among CAR-RNAs, repeat RNA were most responsive to FTO knockout. Repeat RNAs are made up of hundreds to thousands of copies with very similar sequences, Most of them are transcribed from transposons, also known as jumping genes. These genes have the unusual ability to copy and insert themselves in random places across the genome, which can create mutations that may lead to cancer and other genetic diseases. We carefully examined different repeat families and found that LINE1 RNA emerged as the top target. LINE1 RNA itself has the highest basal mCC level and abundance in mouse embryonic stem cells. Upon FTO knockout, it shows the highest number of hypermethylated peaks, the most increased mCC level, and the most reduced expression level. We also validated the changes in LINE1 mCC level and LINE1 expression by RT-qPCR. Together, these results indicate that the primary substrate of FTO in mouse cells could be LINE1 RNA. So we were excited about this. So what is LINE1 and what is LINE1 RNA? So LINE elements are the most abundant mammalian transposons. It comprises more than 20% of human genome. To compare with, only 1.5% of human genome are protein coding genes. The majority of LINE elements is LINE1, and LINE1 is also the only remaining active transposons in humans. It expands by copying and pasting itself to new locations. To do so, LINE1 is first transcribed from DNA into RNA, and then converted back into identical DNA sequences. In addition to the jumping ability, LINE1 RNA can function as a regulatory non-coding RNA. It controls gene expression to modulate self-renewal and pluripotency of mouse cells. LINE1 RNA knockdown arrests the embryonic development at the 2-cell stage. In another study, during the mouse pre-implantation development, perturbation of LINE1 RNA causes severe defects. These studies reminded us of the developmental phenotypes of FTO knockout. So since FTO knockout generally reduces the LINE1 RNA expression in mouse cells, we carefully examined the phenotypic similarities between FTO knockout and LINE1 RNA knockdown. We observed many consistent cellular phenotypes. As example here, both FTO knockout and LINE1 knockdown leads to reduced self-renewal and induced differentiation of mouse cells. These two can be the most important cellular phenotypes of stem cells. More importantly, FTO knockout and LINE1 knockdown shares many similarities in pathway, as exemplified by increased expression of 2-cell stage marker genes and decreased expression of pluripotent genes. These similarities indicate that FTO knockout and LINE1 knockdown may affect similar pathways, supporting LINE1 RNA as a key downstream target of FTO in mouse cells. Therefore, we move on with the focus on LINE1 RNA. We validated the binding of FTO to LINE1 RNA by protein RNA cross-linking and pull-down experiments. Besides knockout, we also treated wild-type mouse cells with a small molecule that can inhibit the demethylation activity of FTO. We also observed the increased mCC level and decreased expression level of LINE1. Furthermore, we designed this CRISPR-based cell-specific mCC demethylation system by fusing DCAT13B with wild-type FTO or catalytically inactive FTO mutant. As we can see here, only deliver wild-type FTO to LINE1 RNA would reverse the effect of FTO knockout on LINE1 mCC level and the expression level. Together, these results establish the causal relationship between FTO-mediated mCC demethylation and LINE1 RNA suppression. So the question we need to answer is how mCC hypermethylation leads to reduced LINE1 RNA level. So the RNA expression level can be roughly estimated by RNA synthesis minus RNA degradation. With high-throughput sequencing, we examined the half-life time and nascent synthesis of LINE1 RNA. So consistent with the general RNA destabilization role of mCC, we observed a slightly faster decay of LINE1 RNA upon FTO knockout. However, LINE1 transcription was dramatically reduced with FTO depletion. We applied the CRISPR system and observed that deliver FTO to long RNA could reverse the effect of FTO knockout, long RNA decay, and long RNA synthesis. We're particularly interested in the transcription part. How could MCC changes in long RNA in turn modulate how it is transcribed from DNA? This is a new feedback on the central dogma. So the activated transcription is generally associated with more open chromatin, which correlates with increased active histone marks or reduced repressive histone marks. So we performed the ChIP-seq to profile the genome-wide changes in a series of histone marks upon FTO knockout. As we can clearly see here, FTO knockout leads to reduced active histone mark H3K4 trimethyl and increased repressive mark H3K9 trimethyl around the genomic loci with M6A marked LAM1 RNA. In addition to the chromatin regulation around LAM1 loci, we also observed the global chromatin closure and globally reduced transcription. In this DNase tunnel assay, cells with more open chromatin are sensitive to DNase treatment and show higher tunnel signals. And in the EU labeling assay, higher EU signal corresponds to faster transcription that incorporates more EU into newly synthesized RNA. So consistently, when treating wild-type mCS cells with the FTO inhibitor, we recapitulate the effect of FTO knockout on chromatin and transcription. Further, we also did the rescue experiments with the CRISPR FTO system. Deliver wild-type to FTO to LAM1 RNA in FTO knockout mCS cell largely rescued the chromatin closure, while delivery of the catalytically inactive mutant shows almost no effect on the chromatin state. This indicates that the chromatin regulation was also dependent on the MCC demethylase activity of FTO. So how could LAM1 RNA demethylation by FTO affect chromatin openness? There are three typical models to potentially explain the role of LAM1 RNA in chromatin regulation. It can be a scaffold for RNA binding proteins, and then recruit epigenetic modifier, transcription factors, and a chromatin remodeler. M6A can serve as a new hub and add another layer to these processes. So I will skip the details of the extensive experiments, but based on this knowledge and our thorough validations, we proposed a model showing the interplay between LAM1 RNA M6A demethylation by FTO and the chromatin state change. So with FTO depletion, LAM1 RNA decayed faster. It cannot maintain the binding of transcription factors and active chromatin remodelers. On the other hand, the increased M6A would promote the nuclear reader YTDC1 binding, and then recruit histone modifiers that can install repressive histone marks. Both pathways lead to chromatin closure. Now we have a general understanding of how chromatin is regulated by this FTO-LINE1 axis. We next asked how this may affect gene regulation. So we analyzed the genes based on their genomic distance to the nearest LAM1 RNA loci. Upon FTO knockout, we found that the expression and the transcription of genes containing LAM1 were particularly reduced compared with genes far from LAM1. We zoomed in on these downregulated LAM1 containing genes, and we found that FTO knockout could also reshape the chromatin state around this LAM1 containing gene loci. We observed the reduced active histone mark H3K4 trimethyl, H3K27 acetylation, and the decreased binding of RNA polymerase II, along with the induced repressive mark H3K9 trimethyl. These results demonstrated the cis regulation of this FTO-LINE1 axis. FTO knockout lead to LINE1 RNA suppression and local chromatin closure, which in turn reduce the transcription of nearby genes. Notably, more than 1,000 genes are subjected to this cis regulation of FTO-LINE1 RNA axis. These are not random 1,000 genes. These genes enrich the key pathways related to cell cycle, cell division, cell differentiation, and early development. This result indicates LINE1 RNA is involved in the aberrant cell differentiation and mouse developmental defects caused by FTO knockout. So with this idea, we re-examined the mouse ES cell differentiation upon FTO knockout. So we can see after differentiation, FTO and LINE1 RNA expression significantly go up. However, knockout of FTO impairs the increase of LINE1 RNA. Again, deliver FTO to LINE1 RNA reverses the effect of FTO knockout on cell differentiation and proliferation. It also reversed the expression of key differentiation markers upon FTO knockout. As a knockout cell with CRISPR rescue and the wild type with control ASO treatment were closely clustered here. Together, these results indicate LINE1 RNA as a major contributor to the phenotype of FTO in mouse cell differentiation and proliferation in an M6A demethylation-dependent manner. Now we know how FTO-LINE1 access works in cell line. We are going back to the question of tissue correlation. We reanalyze the public data. As expected, across human and mouse tissues, high FTO is generally associated with low LINE1 RNA M6A and high LINE1 RNA expression, supporting LINE1 RNA as a major substrate of FTO in these tissues. Notably, brain tissues showed exceptionally high FTO and LINE1 level. Therefore, we took a deeper look at the brain tissues. So in line with what we observed in mouse ES cells, for both cerebellum and hippocampus samples from FTO knockout mice, we observed the increased LINE1 RNA M6A, reduced LINE1 RNA expression associated with dramatic chromatin closure. So in hippocampus, we even observed more than 50% reduction in chromatin accessibility. We further derived the mouse neuron stem cells from wild type and FTO knockout hippocampus. The effect of FTO knockout on mouse neuron stem cells were very similar to mouse ES cells. We can see increased M6A level and decreased expression of LINE1 RNA upon FTO knockout. After differentiation, FTO and LINE1 RNA were upregulated, while FTO knockout impaired the increase of LINE1 RNA. And consistently, FTO knockout led to closed chromatin and reduced transcription in mouse neuron stem cells. So with this data, we show the presence of FTO-LINE1 axis in tissues, especially in brain. So going back to the ultimate question, is FTO-LINE1 axis regulated? Is LINE1 RNA the physiological substrate of FTO during early development? So when we crossed the wild-type male mice with wild-type or FTO knockout female mice, we got much fewer pups for knockout female, and all the surviving heterozygous offspring died in 1 day. These results indicate that FTO knockout in female mice severely impaired the fertility. We therefore studied whether FTO knockout affects the oocyte development. Indeed, FTO depletion reduced the number of GV oocytes by half and significantly reduced the ratio of the healthy oocytes. And consistently, we observed the chromatin closure in GV oocyte from FTO knockout mice. Together, these results show that FTO knockout affects the chromatin configuration of oocytes and can impede their maturation and fertilization. As expected, LINE1 RNA is involved in these defects. FTO and LINE1 RNA expression was strongly correlated during the oocyte development, and LINE1 RNA dropped more than half with FTO depletion in the oocyte. Together, these results clearly shows that FTO-LINE1 axis modulate oocyte development. We further examined the embryonic development. We obtained FTO wild-type embryos and embryos with maternal, paternal, and double FTO knockout. We found that either type of FTO deficiency significantly impaired the implantation rate and the numbers of embryos obtained post-implantation. Notably, we did not obtain any post-implantation embryos upon the homozygous FTO depletion. during pre-implantation development, at the morula stage, where we start to see obvious defects, we found around 40% decrease of LAMA RNA upon FTO knockout, and we also identified many LAMA containing genes that plays essential roles in early development. As example here, we saw significant downregulation of these genes with FTO depletion. So last, I would like to show you the design of this CRISPR-based rescue experiments during early development. So we inject CRISPR FTO with control or guide RNA targeting LAM1 into FTO knockout oocyte, and we fertilize the resulting oocytes with FTO knockout sperms in vitro. After in vitro development, we obtained and examined the resulting moruli. The results are actually beyond our expectation. So deliver FTO to LAMA RNA in oocyte could eventually induce the LAMA RNA expression in the resulting moruli with the dramatically induced chromatin openness and the increased expression of the essential genes that contain LAMA RNA. I believe this was the first case of site-specific MCC demethylation in vivo systems. We thereby established the causal relationship between LAMA RNA, MCC demethylation by FTO with chromatin and gene regulation during early development. To summarize, I hope that I convince you of the presence and functional relevance of FTO-mediated LAMA RNA MCC demethylation in mammals. We show that this FTO-LAMA axis is essential in mouse embryonic stem cells, mouse brain tissues, and mouse early development. More importantly, we discovered that FTO controls the cis regulation of LAMA RNA. So generally, in these primary biological systems, we found that high FTO is associated with high transcription and open chromatin. I think this goes beyond the basic biology. So in the future, whenever we need a transcription burst, we may consider to deliver FTO to achieve it. So we hope to transform what we learn here to biotype. So we already had this exciting case. So overexpression of human FTO in rice lead to expanded roots and increased crop yield. The preliminary analysis show that these developmental phenotypes may also result from the repeat RNA demethylation and chromatin regulation mediated by FTO. So moving forward, we have plenty of opportunities. With this, I would like to thank the support from my advisor and all the colleagues and collaborators contributing to the research work I just talked about. Particularly, Xianbing helped a lot with the bioinformatics part, and our collaborator Wei, Xuekun, Lei, and Xuelian helped a lot with the mouse model. So during the past 9 months, I have also assembled a small but energetic team here in Singapore. So moving forward, in collaboration with the colleagues in chemistry department, we hope to develop biochemical tools to profile and modulate RNA properties. And I also hope to continue the context-dependent mechanistic and functional studies of RNA modifications with a focus on chromatin regulation and small RNA modification. Hope I can share the work from my independent lab with you very soon. And thank you for your attention. I'm happy to take any questions from here.
Thank you for the wonderful talk. So a couple of questions that we got from the audience. So Jessica Willie asked, what do you think is going on with a few genes that are not affected the same by FTO knockout and LINE1 RNA? Do they have something in common? So I think this was slide like 23, 24.
Let me try. You mean the other down-regulated genes, now these overlapped genes? So I think for those genes, they may regulated by the secondary effect of MCC methylation, because we found many transcription factors and other enzymes may be subjected to this down-LAM1 containing gene regulation. So here I show the 3 genes here. This actually, so this LIN28B type 2, G3, GSK3B. These are common downregulated LINE1 containing genes in mouse cell, in oocyte, and in marula. So these genes may have other functions on those downregulated genes not overlapped with LINE1 containing gene.
So the other question, which is also my question too, is how is the specificity conferred by FTO recognizing LINE1 RNA compared to all the other M6A RNAs?
So I think there is a study of FTO binding partners that may help FTO recognize substrate. So there's one partner gene named SFPQ. So this protein is a LINE1 splicing regulated genes. So maybe this gene can help FTO to recognize LINE1 RNA. And also FTO distributes differently in different biological systems. So I think in this primary tissues, FTO just localized near those LINE1 transcripts.
Got it. Thank you for the wonderful presentation. And so we're going to move on to the next speaker. So it's my pleasure today to introduce Dr. Huili today. She graduated from the University of Cambridge in 2005 with a BA degree in Nature Sciences. She then went on to complete her PhD at MIT in David Bartel's lab, where she made contributions towards our understanding of microRNA-mediated repression, which was recently awarded with the Nobel Prize. Huili was an independent fellow at IMCB from 2012 to 2021. She has earned significant awards, including L'Oréal Singapore for Women Science and the Young Scientist Award. She's now a senior research scientist at the CSI Institute of Singapore, and we're really excited to have her here. So today she'll be sharing her work on ROS-induced translational regulation through spatiotemporal differences in codon recognition is a key driver of brown adipogenesis. So please welcome Dr. Guo today. Looking forward to your presentation.
All right, thank you very much, Anthony, and thank you for the opportunity to present at the RNA Collaborative series. I'm going to tell you a story today on how ROS-induced translation regulation can be a key driver of brown adipogenesis through differences in codon recognition. All right, here we go. Okay. So we went into this simply wanting to understand what it takes to make mitochondria, because we know that you need more than 1,000 protein components to make mitochondria, but only 13 protein coding genes are encoded within the mitochondrial DNA itself. So for the rest of the proteins to be expressed and then assembled into mitochondria during biogenesis, there must be a lot of coordination. Traditionally, this is much better studied at a transcription level. So our question was just, what is the translation coordination that is involved in this process? So to this end, we made use of a brown fat differentiation model. Because when you induce pre-adipocytes to mature brown fat cells, they undergo massive mitochondrial biogenesis. So this is a 6-day process where we would harvest the cells every single day of differentiation, and then at every single day we would perform this workflow. So there is the bulk cytoplasm, which you can think of as the whole cell lysate without nuclei, and then we would fractionate the lysate into the crude mitochondria fraction as well as the rest of the cytosol. And to each compartment, what we would do is in parallel RNA-seq and ribosome profiling. So we can quantify the translation efficiency for all the genes that are sequenced. And at the same time, because we have sequence information from the crude mitochondrial compartment and the cytosolic fraction, we can derive an enrichment ratio where if there were some mRNA that were more enriched around the mitochondrial vicinity, we would be able to identify them. All right, so what does the data look like? Okay, so you're looking at volcano plots from days 1 to 6 relative to day 0 before induction. And we have translation fold change on the x-axis. And so genes over here are upregulated translationally and genes over here are downregulated. So you can see that at the whole cell level, translation regulation can be seen from day 4 onwards. And the mitochondrial components are colored brown and they skew right because obviously the cells are massively upregulating mitochondria during this differentiation process. In the crude mitochondrial fraction, if you were to do the same analysis to the exact same genes, what you see instead is that you get translation regulation from day 2 onwards. That's 2 full days before day 4. or which is what you can see at the whole cell level. Now, why is it that we don't see translation regulation at day 2 at the whole cell level? This is because this translation regulation in the mitochondrial vicinity is masked by the rest of what is happening in the rest of the cytoplasm, which is there's not much translation regulation going on at all. So again, exact same genes, same analysis, but from the cytosolic fraction for this row of volcano plots. So not many genes are above the line of significance here, indicating not much translation regulation going on in the cytosolic fraction. Now, we wanted to ask if there was any relationship between the translation regulation that we see and the frequency of codons in mRNAs. So we made these scatter plots where you plot translation efficiency for change on the y-axis and then the frequency of a particular codon on the message open reading frame on the x-axis. So for example, this gene over here has lots of GUG in its coding region, and then this gene over here has fewer GUGs. And because you get a correlation coefficient that's positive, that means the more GUG you have in your coding region, the better translated you are over time during this differentiation time course. Now, if we can do this for one codon, then we can plot this scatter plot for 61 possible coding codons. But in order not to look at 61 scatter plots, we just simply took the correlation coefficient and plotted it as a bar. So this is for GUG in the previous slide. Then we get this consolidated bar plot where you see a hierarchy of relationship between translation regulation and codons. So I'm going to be showing these this kind of plot in the next slide. Okay, now here we have, by the way, don't worry if you can't read the codons, you're not meant to be able to read the codons. Okay, now we have group mitochondrial fraction data, plotting these consolidated bar plots from day 1 to day 6. And again, all the translation fold changes are relative to day 0. And what I've done here is I've colored all codons ending in G or C green, and then codons that end in A or U are colored purple. So I think we can all appreciate that all the green codons are bunched up together on the left-hand side of the plots and on the positive end, meaning the more G and C ending codons you have, the better translated you are during this differentiation process. Now, the situation is kind of the opposite in the cytosolic fraction, where in the beginning of differentiation, it's the purple codons that are on the positive end. And then somewhere at day 4, there's a switchover. And then by days 5 and 6, you have the same scenario in the group mitochondrial fraction. So it appears that there's a translation bias towards mRNAs with more G and C ending codons. And there is a signal that starts off from the mitochondrial vicinity that later spreads to the rest of the cytosol. So we didn't just see this bias happening to 1 or 2 amino acids, because if you see a bunching up of all the green codons, this is an across the board translation bias towards essentially all G and C ending codons, right? So a shift towards G and C ending codons, which is the third base of the codon, i.e. the wobble base, suggests that there might be changes in the tRNA pool within the cells. So we looked at tRNA abundance in the 2 compartments and didn't see differences that could explain this large-scale translation bias that we see between the 2 compartments. So that leaves us with tRNA modifications. tRNAs are known to be extensively modified, and modifications to tRNAs in the anticodon region or in the surroundings can lead to changes in the way the tRNAs recognize their codons. So knowing this, that there's a large-scale translation bias towards all G and C ending codons, we searched the literature extensively for something that can explain this phenomenon, and eventually landed with this enzyme called ERP3, which is also a histone acetylase, so it's in the nucleus, but it also works in the cytoplasm where it modifies 11 tRNAs and hence can affect the translation of 22 codons. So what ERP3 does is that for tRNAs affected by ERP3 modification, they have a uridine at position 34, and this recognizes the wobble base over here, either the A ending one or the G ending one for the same amino acid. Now, if ERP3 modifies this uridine, then the tRNA now prefers to recognize the A ending codon. And ERP3 itself is a rate-limiting enzyme, and it adds the CM5 group to the uridine. And subsequently, other enzymes add other modifications, and eventually you end up with these 4 final modifications affecting 11 tRNAs and hence the translation of these 22 codons. Because it's 22 codons, so essentially if there is just a change to the activity of ERP3, it could possibly explain all of the G bias that we see, i.e. the translation bias towards mRNAs with G ending codons. Okay, so in our case, because we are seeing as we differentiate the cells from pre-adipocytes to mature brown fat, a G bias, right? So for us, the direction should be going this way, i.e. we should see a reduction in ERP3 activity, because when you go from a preference for A ending codons to no preference for A ending or G ending codons, then you essentially get an apparent translation bias towards G ending codons. Right? If that is the case, this would predict that if we were to profile tRNA modifications by mass spec, then look at the ERP3 mediated modifications, then we should see a reduction in modification levels over time. So we purified tRNAs from the crude mitochondrial fraction and the cytosolic fraction and looked at the ERP3 mediated modifications over time. So you can see the tRNA modification levels mediated by ERP3 drop over time, and the drop is more pronounced in the crude mitochondrial fraction and occurs earlier, which is consistent with us seeing a larger translation bias and earlier bias in the crude mitochondrial fraction. Turns out that this ERP3 enzyme is an iron sulfur cluster containing enzyme, meaning its activity is likely to be influenced by reactive oxygen species, because the ROS can change the oxidation state of the iron in the iron sulfur cluster. So that gives us this working hypothesis, which goes like this. Before differentiation, you have all these modified tRNAs. And then when you induce the cells to differentiate, the initial mitochondrial biogenesis would generate ROS as a byproduct. And that would lead to a reduction in ERP3 activity in the vicinity of mitochondria and raise the proportion of unmodified tRNAs in the region, producing an initial translation bias towards G ending codons, first in the mitochondrial vicinity. Eventually, as your cells differentiate further, and you get more and more mitochondrial biogenesis and more ROS, this phenomenon spreads through the rest of the cell. In data that I don't have time to show today, we found that ROS is absolutely required for brown adipogenesis. And as you would expect, when you differentiate the cells to become mature brown fat, the ROS levels increase because of massive mitochondrial biogenesis. So this reduces the activity of ERP3 and hence ERP3 modifications, as we saw by mass spec, and in turn produces a translation bias towards G ending codons. Now, at this point, so far, this translation bias that we observe has been inferred from sequencing data, i.e. We saw more ribosome footprints from the ribosome profiling in mRNAs, on mRNAs that have more G ending codons. Now, however, for us to get more ribosome footprints, you can either have more translation, or if your ribosomes are stalled or moving slower, you can also get more ribosome footprints, right? But without an actual increase in protein synthesis. So to distinguish between these two possibilities, we made use of luciferase reporters. We took firefly luciferase and then changed the third base of the ERP3 affected codons. If they didn't end in a G, we would swap the base to G ending to get a G maximized luciferase, and vice versa to get an A maximized luciferase. Now, this is the wobble base, so the amino acid doesn't change. This is still firefly luciferase, but we would expect the G maximized luciferase to be translated better than the A maximized one if our hypothesis is correct. Okay, so we differentiated the cells and transfected in these luciferase reporters and saw that the G maximized luciferase is translated better than the A maximized luciferase. And this model also predicts that if we were to reduce ROS levels, then the G bias would decrease. So we put in Thyron, which is an ROS scavenger, to deplete ROS from the system before differentiation, and then did luciferase assay at the end. The G bias didn't just decrease, but the translation bias shifted instead towards A ending codons when ROS is depleted, so which is consistent with our model. All right, so it's all well and good that we identified an interesting phenomenon where from initial differentiation induction and ROS production, you get this translation bias towards GN encodons. But how does this matter to the cell? And it will matter to the cell if the mRNAs coding for brown fat or mitochondria-related proteins are benefiting from this G bias, because then that would push the cells towards further along the differentiation pathway. So to address this, we took a different approach. We ranked all the genes according to whether they overuse GNC N encodons or underuse GNC N encodons for the same amino acid. So if these brown fat differentiation pathway related genes and these mitochondrial components are found here, then they would be expected to benefit from the translation bias towards GNC N encodons and hence drive brown adipogenesis. So indeed, we found that these two brown fat differentiation regulators and mitochondrial membrane components do overuse GNC N encodons. And thus, they would benefit from the translation bias, and this would drive brown adipogenesis. Another group that we didn't quite expect, but that we found that also would overuse GNC N encodons were the ROS scavengers. So then this gives rise to an interesting idea, which is that once you induce the cells to differentiate and you get initial mitochondrial biogenesis, you start to increase ROS levels in the cells. This would reduce ELP3 activity and hence ELP3 mediated modifications on tRNAs, producing a translation bias towards GN encodons. Brown fat differentiation regulators and mitochondrial membrane component encoding mRNAs benefit from this bias, thus driving brown fat differentiation. But at the same time, as differentiation progresses and you get more and more mitochondrial biogenesis, you inevitably increase the level of ROS in the cells. So how do you have enough ROS to kick off this pathway, but at the same time don't produce so much ROS as to kill the cells, right? So if the translation of ROS scavengers also encoding mRNAs also benefit from this G bias, then this could be a way for the cells to exert negative feedback to minimize collateral damage from having too much ROS, and it might enable a form of sustained mitochondrial biogenesis. At this point, there is also the question of what is the proportion of expression increase that is actually driven by the translation upregulation, right? So if there is a translation bias, but it only say increases, only contributes to the overall expression increase by 5%, then we can't really say that this translation bias is driving brown fat differentiation. So we looked at I'm going to show you an example using these mitochondrial proteins encoding mRNAs. So like I said before, mitochondrial membrane components tend to be on this end of the spectrum. They overuse GNC N encodons, whereas mitochondrial matrix components tend to underuse GNC N encodons for some reason. Okay, so if we compare different groups of mitochondrial membrane components against the matrix components, which show the opposite, have the opposite characteristics, then you can see how much translation regulation is contributing towards the overall expression increase. So the first group here is mitochondrial import proteins. So what you're looking at is median log4 change in RNA-seq or translation efficiency plotted on the y-axis against days of differentiation. And the brown line depicts this log4 changes in the crude mitochondrial fraction. The pink line is for the bulk cytoplasm, i.e. Pretty much whole cell. And then the blue line is cytosolic fraction. So you can see that the RNA-seq changes and translation efficiency changes are pretty much comparable, meaning that translation regulation, this translation bias is contributing a substantial amount towards the overall expression increase. So this is mitochondrial import proteins, and then respiratory ECC complexes and metabolite carriers. These are all mitochondrial membrane components. And there is a very characteristic boost in translation on day 2, which is exactly when we saw this translation regulation happening in the mitochondrial vicinity in the earlier slides for mitochondrial membrane components here. Okay, and this they contribute at least 50% towards the overall expression increase. Now for the matrix components, example, fatty acid oxidation enzymes, enzymes in the TCA cycle, mitochondrial processing peptidases, the expression increase is driven more by increases in mRNA levels. Nevertheless, translation regulation still contributes about 30% towards the overall increase. So in other words, not insubstantial. All right, so this model still stands, and we think that this has interesting implications for other contexts in which mitochondrial biogenesis and ROS levels play an important role. And with that, thank you very much, and I'm happy to take questions. Thank you.
Thank you for the wonderful talk. So I'll start with one question from Jian Schaffer. Is your luciferase part of studies for testing codon bias? Did you test both cytosolic targeted and mitochondrial targeted constructs?
I didn't target the luciferase reporters. But that's the thing that we are currently testing. Basically, so we didn't modify the luciferase reporters besides the coding region, so we don't expect them to be targeted anywhere in our current in the slides that I showed. But now we're trying to localize the luciferase reporters to the mitochondrial vicinity and see if we get a larger effect earlier.
So I have a separate question. Do you see any effects on RNA stability with this GC codon bias?
We didn't test stability per se. So besides abundance, RNA abundance, I won't be able to answer the stability question. Yeah.
I see. And do you see in other models of increased mitochondrial mass can switch on translation of genes that are targeted to mitochondria?
Okay, so there are a bunch of studies in yeast where this ELP3 mediated selective translation can help to regulate stress responses. In other words, if you stress the cells, you can the codon bias that's produced preferentially translates mRNAs that end up to mediate stress response and recovery program. But those are not mitochondrial, as in mitochondria are not intimately involved per se. So and then there are also other ones where you have ELP3 mediated codon bias, similar to what I'm seeing here, but biasing macrophage polarization, right? So in the immune system, and then others in the neuronal system. But I haven't seen something that is describing mitochondrial biogenesis alone. So this is something that we are planning to test in other systems like muscle differentiation.
In your model, you talk about how it's like spatially restricted in the first place and then it'll spread. So what's keeping it in like spatially restricted? I imagine tRNAs would just fly around in the cell because they're pretty small. So what keeps this whole like translation spatially restricted? Bias do you see near the mitochondria? Do you have any idea?
Yeah, I didn't touch on any of the localized mRNA translation in this talk because there isn't enough time. But we do have we do have indications of localized mRNA translation because we did do RNA-seq and ribosome profiling in different compartments. And then, so definitely for the mRNAs that are attached to the surface of mitochondria, and there are such mRNAs, we can see that the mRNAs found in the vicinity of mitochondria, or maybe you can think of it as surface attached, they tend to be enriched in G and C ending codons in the first place. So even before you start to differentiate the cells have already partitioned the mRNAs in such a way that the mRNAs that end up benefiting the most from the translation bias are already there next to mitochondria. Yeah, I don't know why, but
I see. So there's no mechanism why these mRNAs are targeted there, just based on this GC, or they could be.
Yeah, I don't know the mechanism.
Okay. Very cool. Thank you for the presentation.
Thank you.
I think we have a couple more minutes. Anyone has any other questions for both speakers? All right, thank you so much. So the next RNA Collaborative Seminar Series is on October 23rd, hosted by OSU Center for RNA Biology. So looking forward to see you guys all there. All right, thank you so much.